So welcome everyone to our next webinar series. And today is my great pleasure to introduce the first speaker, Jonathan Ostrem. So Jonathan is an assistant professor at the Department of Medicine at the UCSF and an oncologist at the Helen Diller Family Comprehensive Cancer Center. He obtained his MD and PhD from the UCSF working with Kevin Schockett. And after the clinical training at the Brigham and Women's Hospital and the Dana-Farber Cancer Institute, he completed a postdoctoral work with Dr. Stuart Schreiber at the Broad Institute of MIT and Harvard. And today, he will share with us a truly fascinating story about small molecule being ubiquitinated. Jonathan, thank you very much for joining us, and we are very much looking for your seminar.
Thanks so much, Nikolai, and thanks everyone for being here. It's really exciting to be a part of the DPD webinar today. It's such a great series. So I'm excited to tell you about this story that started at the Broad Institute and at Dana-Farber, and that I've continued in my own lab about the serendipitous discovery of direct small molecule ubiquitination. In this project, I really got into this project because of the fact that we all realize that small molecules can do much more than inhibit or activate proteins in the cell, that they can do things like bring proteins together, change their interactome in the form of molecular glues, or transfer post-translational modifications from an enzyme to a substrate. Things like molecular glue degraders. This audience obviously needs no introduction to these things, but the reason that I bring this up is because some of the most fascinating mechanisms of small molecules have come from phenotypic screens identifying small molecules and then working to understand their mechanism within the cell and finding completely novel mechanisms this way. And that got me interested in this set of molecules, exemplified by the molecule in black here, BRD 1732, that was found from a phenotypic screen. And the reason that I thought, my interest was initially started was that it's highly stereospecific in its cytotoxicity. So you change one stereocenter, it has 3 stereocenters, and you change any one of those stereocenters, and the compound becomes dramatically less cytotoxic, shifting the IC50 almost 100-fold. Shown here in the multiple myeloma cell line, MM1S, you can see in blue and red, the enantiomer and diastereomer are dramatically less cytotoxic, or the IC50 shifts. To try to understand the mechanism in cells, we used this powerful platform at the Broad Institute called the Proteasome platform, which is a panel of bar-coded cancer cell lines that have been highly characterized at the transcriptional level, proteomic level, mutational level, lineage, and it's possible to screen a perturbation of interest and pull out the features that correlate either with sensitivity or insensitivity. And when we did this for our compound BRD 1732, what came out is high expression of this gene RNF19A correlated with sensitivity to our compound. And turns out RNF19A is an E3 ubiquitin ligase. When we then performed a genome-wide CRISPR screen in collaboration with Konstantin Mitsiadis' lab, we found not only that knockout of RNF19A confers resistance, but knockout of its paralog, RNF19B, also confers resistance, as well as knocking out UBE2L3. Turns out UBE2L3 is the E2 ligase most associated with these 2 E3 ligases. This is, again, performed in MM1S cells. Moving into a different cell line, however, looking at just HEK293 T cells, we made a double knockout and showed that confers resistance, knocking out both RNF19A and RNF19B confers resistance. And if we then re-express RNF19A, we're able to reinstitute the sensitivity to the compound. So again, this is just where these sit in the cascade of ubiquitination. And I think with this audience, everyone is obviously expecting the next slide to be quantitative proteomics, but I'm not going to show you just yet. What we after pursuing for a long time, trying to identify what the neo-substrate was, and that this was going to be a new molecular glue degrader, and being unsuccessful in those efforts, we were looking at general effects on ubiquitin in the cell, and noticed this curious phenotype where when we treated cells with 1732, we saw what looked like an increase in monoubiquitin, as well as these unanchored polyubiquitin. I'm just showing you diubiquitin here. So you can see monoubiquitin has increased and this new diubiquitin band. And it turned out that that effect went away if we knocked out RNF19A and RNF19B. And we could recover that phenotype by re-expressing either RNF19A or RNF19B. That effect is also stereospecific. So I'm showing you here the main compound, BRD1732, with the dose response effect. That effect is shifted if we use the enantiomer. And after performing many, many replicates of this sort of experiment in various conditions, we noticed that it seemed like monoubiquitin in treated cells ran at a slightly higher molecular weight than in untreated cells. I don't know that I could convince you of that with this plot here. Maybe if we zoom in, you can see that this darker band looks ever so slightly higher. And to try to convince ourselves of that, we ran this ubiquitin blot on a high percentage gel. This is now untreated, treated, untreated, treated, and you can see there's actually a true shift in the molecular weight or in where the band for monoubiquitin runs on this gel. And so we came up with a very simple purification of ubiquitin from cells. And you can see here, take just XB293 T cells, these specialized HEC cells, sonicate them, run over cation exchange, and then size exclusion. And this is monoubiquitin from untreated cells that comes out. You can see here the expected molecular weight of 8565 Daltons. When we pre-treat cells for 6 hours with our compound 1732 and do the same thing, you can see that ubiquitin in the cell now has a new molecular weight of 8952. And to our surprise, that exactly matches the expected molecular weight of a conjugate between ubiquitin and our compound. So our ubiquitin plus our compound minus water. Now, it turns out if you take ubiquitin and you don't denature it, and then you treat it with trypsin, the primary cleavage site is after residue 74, meaning that you've only cleaved off the terminal diglycine of ubiquitin. So we did this on ubiquitin purified from 1732 treated cells and ran that on a mass spec. And you can see here this envelope in red here that deconvolutes to the first 74 amino acids of ubiquitin. And what we saw is this new peak showing up, 519. And when we performed MS-MS fragmentation on that, first thing that came off was the molecular weight of our compound, BRD1732, as well as many of the fragmentation ions from 1732 fragmentation. So this suggests that just in the way that ubiquitin transfers from either an E2 or E3 ligase to a lysine on a substrate protein, our compound is acting akin to a protein and getting directly ubiquitinated, transferred to the C-terminus of ubiquitin itself. Now that brought to mind the story of these Takeda compounds, originally Millennium, MLN 7243 or TAC 243, that acts, it's an AMP analog. It binds to the E1 ligase and acts as an AMP analog to get added to the C-terminus of ubiquitin, similar to our compound. And so we wondered, is 1732 acting as an E1 inhibitor? And maybe that's the mechanism of cytotoxicity. Initially, we started with just a biochemical assay, purified E1 and E2 enzymes with ubiquitin in our compound. And what you can see in this assay, where we're monitoring for ubiquitination of the E2, we see efficient ubiquitination of the E2 in the presence of 1732. So it's definitely not acting in the same way as MLN 7243. But we wondered, what about in cells after the compound has been ubiquitinated? Can it then circle back and inhibit the E1 ligase? And so this is now cell lysates, where we've treated with a range of concentrations of our compound 1732, monitoring for this ubiquitin phenotype, but also monitoring for polyubiquitin levels in the cell. And you can see there's very little effect on the accumulation of polyubiquitinated species in cells on treatment with 1732, whereas when we treat with the E1 ligase inhibitor, at increasing concentrations, we see depletion of these polyubiquitinated species as we shut down the ubiquitination cascade. Now, we noticed that this monoubiquitin species and the diubiquitin species are actually accumulating to quite a dramatic degree at increasing concentrations. This is now up to 10 micromolar. We just see a truly dramatic accumulation of this monoubiquitin species. And with such dramatic effects on ubiquitin in the cell, we wondered whether we're affecting proteasome function. And to try to answer that, we used the classic system of lenalidomide-induced ubiquitination and degradation of IKZF3, which is cereblon-dependent proteasomal degradation. We used this reporter system from Ben Ebert's lab, which is IKZF3 fused to GFP with an internal ribosomal entry site, followed by M-cherry. And in here, down here, I'm showing you unlabeled cells in gray, the reporter cells here in dark gray, showing GFP expression. When we treat with lenalidomide alone, we see a reduction in GFP, which corresponds to the degradation of IKZF3. If we co-treat with the proteasome inhibitor MG132, we completely block this effect. However, if we co-treat with our compound 1732, we see essentially no effect on loss of GFP, so essentially no effect on the degradation of IKZF3 by lenalidomide. However, if we pre-treat cells for 6 hours, now we see a complete blockade of degradation of IKZF3, suggesting that either there must be an accumulation of something, so perhaps this is correlating with accumulation of the ubiquitin 1732 conjugate or depletion of something. This is an exogenous system, and so we wondered, what about an endogenous system of degradation? And that brought to mind TNF-alpha induced degradation of I-kappa B alpha. It's known that upon treatment with TNF-alpha, I-kappa B alpha gets first phosphorylated and then ubiquitinated and sent to the proteasome to be degraded. And so here on the right, I'm showing you a Western blot demonstrating just that in wild-type cells. If we treat with TNF-alpha, we can see loss of I-kappa B alpha. If we pre-treat for 6 hours with BRD 1732, we completely block that effect. If we look at RNF19B knockout, and then we also perform siRNA for RNF19A, we see no effect on TNF-stimulated degradation in the absence of our compound. But now we completely lose the effect of 1732 in the combined knockout and siRNA. And it's important to say that if we treat for only 1 hour with 1732, we see no effect on TNF-stimulated degradation by I-kappa B alpha, whereas MG132 completely blocks the effect with co-treatment or 1 hour pre-treatment. So that raised the question, well, what's happening with other endogenous proteins within the cell? And so we took a set, on the left here, I'm showing you a set of proteins with a short half-life that are known to be degraded by the proteasome. You can see if you treat cells with MG132, these proteins accumulate after 6 hours. If we treat cells with 1732, also for 6 hours, we see that they accumulate. And what's interesting is that we see this alternative banding pattern above many of the proteins that's distinct from MG132. And it turns out if we co-treat with MG132, that phenotype reverts back to what looks like MG132. We performed quantitative proteomics at 24 hours, comparing our compound 1732 with MG132. And what we see is that there are actually a striking number of proteins that are increased by both that are not changed at the transcriptional level. But there are also quite a few shown in red here that are distinct between these two compounds. And what was very striking is that on treatment with 1732, not only did we see stabilization of a wide range of proteins, but we actually saw destabilization of a significant fraction of the proteome. This is a very late time point, and a lot could be happening. The cells are probably starting to undergo apoptosis, and so we performed a time course looking at earlier time points. And now this is the reason that I didn't lead with the quantitative proteomics before, because you'll see that at 4 hours, we see quite profound loss of these two zinc finger proteins, ZFAN5 and ZFAN6. These are not known to be substrates of RNF19A or RNF19B. Here in blue, I'm showing you what are reported to be substrates of RNF19A and RNF19B, and we can see that they're not actually dramatically changed at 4 hours. ZFAN5 and 6 are zinc finger proteins. They have two zinc finger domains, an N-terminal A20 domain and a C-terminal AN1 domain, and they're actually highly homologous. Within the A20 and AN1 domains, they're greater than 80% sequence identity. What was interesting to us is that their loss, on the one hand, is prevented by co-treatment with a proteasome inhibitor, MG132. So this is true degradation by the proteasome. But what you notice here is that if we co-treat with an E1 ligase inhibitor, MLN7243, degradation is not prevented, meaning that this is ubiquitin-independent proteasomal degradation that's induced by 1732. Now, you'll notice also that MLN7243 itself induces some degree of degradation of ZFAN5 and ZFAN6. So that was curious to us. Now, ZFAN5, it turns out, actually directly binds the proteasome regulatory cap, the 19S proteasome. This was shown in an excellent paper by Donghoon Lee at Harvard. And it's known that ZFAN5 from these studies actually enhances proteasomal degradation of ubiquitinated substrates. And because we were seeing this similar degradation by 1732 and MLN7243, we wondered if this effect on ZFAN5 is really because ZFAN5 is monitoring the ubiquitination state in the cell. We're continuing to work on understanding the mechanism of degradation, but our current model of the effect of 1732 in cells is that it enters the cells, it undergoes ubiquitination by UBE2L3, followed by RNF19A and RNF19B. Upon ubiquitination, as it accumulates at very high concentrations, it's able to inhibit proteasomal degradation of ubiquitinated substrates while having no effect on proteasomal degradation independent of ubiquitin. Now, these data are consistent with the fact that 1732 blocks ubiquitin-dependent degradation of ICAP-B alpha or IKZF3, but does not block the ubiquitin-independent proteasomal degradation of ZFAN5 and ZFAN6. Also in our TMT data, we follow ODC1. Now, ODC1 is known to undergo rapid constitutive ubiquitin-independent degradation, and we don't see stabilization of ODC1 on treatment with 1732. Because we've now demonstrated that BRD-1732 can undergo ubiquitination, this obviously directs toward a very exciting possibility that we could induce the degradation of a protein through a ubiquitin-dependent pathway, but instead of direct ubiquitination of the substrate protein, that ubiquitination could occur through a small molecule mediary, such as BRD-1732. As additional kind of suggestion in that direction, although we haven't been able to prove that yet, we see not only monoubiquitination of BRD-1732, but polyubiquitination. So far, in several cell types, we've seen upwards of 4 ubiquitin molecules put onto BRD 1732. We've confirmed that the linkage of that polyubiquitination is K48. And so I think that there's a lot of promise here for the possibility of polyubiquitination of a small molecule to direct the degradation of a protein. Just to summarize, we demonstrated that this molecule, BRD 1732, gets directly ubiquitinated in cells, can accumulate to a dramatic degree. We do know that it's getting ubiquitinated at much lower concentrations at which the proteasome is not inhibited. And so I think that still lends itself toward the possibility of this small molecule mediated degradation of proteins. RNF19A and B are essential for both the cytotoxicity and the ubiquitination, although we have not been able to reconstitute the reaction in vitro. We're still working on that. I'm quite hopeful that we'll get that soon. And then, as I've said before, I think this suggests an entirely new mechanism of small molecule directing post-translational modification through the small molecule itself. With that, I'd like to thank everyone involved in this work, in particular, the phenotypic screen, and a lot of the early biochemical work was done by a graduate student that was joint between Stuart Schreiber and Sigal Kadosh. His name's Enrique Garcia Rivera, and then a truly exceptional postdoctoral fellow in my lab, Wei-Chung Lee, who has done a lot of the key work demonstrating this direct ubiquitination in cells. You can find the work deposited here on bioRxiv here, and thanks to funding mechanisms and all of our collaborators. And I'll take questions. Thank you.